I'm housed in the bioengineering department, and I'm a postdoc there. And I just wanted to give a disclaimer that I am by no means a trained digital artist. And if you have any experience with this, I hope that it's not frustrating for you, but I'm just going to give you my experience designing using 3D animations and how I think they can be useful. But I'm completely self-trained here. So, I'm just kind of giving you an idea of how these things can be used. But I think, what I hope to get across is that you can teach yourself how to do these things just simply by going online and looking at tutorials on YouTube and things like that. Because my background is mostly in bioengineering and microbiology. So I have a deep understanding of these things I'm going to show you, but I've recently learned how to use these animation tools. So I think, first question you should ask yourself is, when should you use 3D animations? And just to give you an idea what this presentation is going to look like, I'm basically going to be showing you some examples where we've used 3D animations and some cool tools that are very useful to help describe your research. So that's why I'm kind of asking this question: when is it appropriate to use 3D? And I think, Adobe Illustrator is a great tool, or even PowerPoint, for making most of our animations and describing our research. But sometimes it can be useful to make 3D animations. And Alice Kitterman, who's an illustrator at Science, actually has this article, "To 3D or Not to 3D." And she very beautifully shows here like what you get by creating 3D animations. So again, this is not going to be quantitative things we're talking about today, but more how to represent your research. But what she's showing here is this macrophage kind of attacking this cancer cell. And you can appreciate that with this 3D animation, you can better appreciate the cell-cell interaction that's going on. It's quite dynamic, and there's this whole three-dimensionality to it that would be lost with 2D animations. And Alice has this nice flow chart that kind of helps you identify when you should use 3D animations versus 2D animations. And really what it comes down to is, if what you're working on is this complex idea, 3D animations are probably going to be useful because there's a lot of things going on, but it's a lot easier for the human eye to conceptualize things that are 3D, even if they are very complex, because that's the world that we're used to perceiving as 3D. So I think it really helps drive home the idea a lot better if you can make it in 3D. But a lot of times you won't have time. You might just be doing it because it looks pretty. That's not really a good enough reason. Or you just want to try it. That I think that's a good enough reason, but Alice kind of said, maybe you shouldn't, and you should just go towards 2D animations. But I think she has a beautiful example here showing these molecules in this 2D representation. But if you make them in this 3D schematic, you can really appreciate how these molecules can interact and the conformational changes that can occur. And there's no good way to get that across using 2D animations, or as well as like these gear-like molecules that can rotate around one another. 2D animation or 2D renderings don't really do that justice. So next I'll kind of get into how I got into 3D animation, so that hopefully it helps you take that step as well. So back in fall of 2019, we were lucky enough to have a paper accepted, and they invited us to submit some cover art for our publication. So this was based on some collaborative work with the Chenoy lab, where we had created these fibrous hydrogel materials that would actually plastically deform and form new bonds whenever forces were exerted on them. That's what I'm showing here. So it was a very cool material system that we worked on. And I wanted to really accurately represent this. And I could have reached out to an artist, or I guess that was our one option, is to reach out to an artist to make this, but artists are notorious for not really being able to conceptualize what your research looks like. And it's a lot of trouble to work with them. And it also costs a lot of money just to submit these cover arts. So I took it upon myself to learn a 3D animation software. And within a couple of weeks, I was able to make this rendering here that they accepted for the cover of Advanced Materials. So if you have any experience using CAD software, it was very easy to just jump right in and start making these things. And what's beautiful about these softwares is that they can kind of render your CAD um, um, design and make it look really nice without you having to do much work. So the softwares really do a lot of the work. So here's some examples of some 3D animation software. Maya is one that I've never used. Blender, I highly recommend Blender. It's completely free. It's not as intuitive and user-friendly, so that's why I haven't used it as much. So it's not as easy to get going, but there's tons of tutorials and plugins online for using this software. So I would suggest if you're starting out fresh to use Blender. We've even used it to like simulate particle tracking and things like that. But today I'll be talking about Cinema 4D, which is also free for academics. And it has a very user-friendly interface, and that's why I like it. And it also has this really nice renderer. So once you make your design, it can just create these beautiful images of what you've designed. So like I said, to make this more approachable, I'm just going to show you guys literally what it looks like working in one of these softwares. And then throughout the presentation, I'll show you some other aspects, some animations and where we have applied 3D animations, some useful plugins, and last, some sculpting that you can do. It's more of an advanced technique, but I think it's super useful for the things that we like to visualize. So this is a typical interface for one of these animation softwares. This is Cinema 4D that I'm showing here. But as you can see up here, there's a bunch of different tools and it's probably a little intimidating. But I highly suggest just downloading one of these softwares and clicking around, and you'll quickly figure out how to start making some shapes and maybe even things that are relevant for your research. So I'm not able to actually go into the software. This is just a recording of me using the software because it would probably crash my computer if I was trying to use Zoom at the same time. But we can go into this shape tool here, and we're going to pick a cube. And you literally just click on it. It adds this cube to 3D space, and we can rotate around it just like any CAD software, although it's a lot simpler than most CAD software because you're just kind of selecting shapes and it's less quantitative about what the dimensions are and things like that. But you can see this is just made of single panels on the side. If we want to add more voxels to this object, we can add them. So here I've created a 10 by 10 by 10 voxel of this cube. And then another useful, so that kind of adds more dimensions to this object. And now another useful tool is this cloner tool. So all of these animation software will have this cloning tool where you can just iterate and create a lot more of whatever object you've created. And this is useful for kind of building up and filling 3D spaces for your renderings. So we'll kind of separate these out and create this array of these cubes here. So it's very quick. You can just add in how many you want and the spacing of all of that. And next we're going to add a color to it just to give it a little more flair. So we'll just pick this green color and we drag that onto the cloner. And now we have these green boxes. We're going to create a floor now that we can drop these boxes onto. And this is another really useful aspect of these kind of softwares is that you can simulate gravity. So I'm adding this kind of collider tag to this plane here that I'm acting as kind of a surface that these cubes are going to collide with. So I added the rigid body tag, and as I hit play here, what's going to happen is that these objects are just going to fall due to gravity. And they all kind of stack on top of each other. So to get something that's a little more interesting, we can create like different radial arrays of cubes. And as we drop those, we can see they're starting, that looks a little more interesting. And we can clone them around other objects like this sphere that I just showed. So if we get rid of the sphere and then we just have these cubes kind of floating in space, we can then look at these, falling. And this is just a little more interesting to watch. So again, simulating gravity is a very useful way to kind of let the software create something that will look more relevant. But I haven't talked yet about the rendering aspect of this. So we can create things, we can put them in 3D space, make them look kind of relevant to maybe your research. But I think one of the really cool things about these softwares is that you can play around with light. And that's not really possible with 2D renderings because you're not going to have a three-dimensional object that will cast shadows. And that's what really sets these softwares apart. So here I'm introducing an area light that will just kind of cast light everywhere. I'm putting it in the center of this sphere, and you can get this kind of cool, where there's like a light coming from inside and lighting up all these surfaces, but the outside are shadowed. And then what we do is we just go to this play sign here. We hit that and render to picture viewer. And then it's literally just going to render this in a nice rendering software here, and we get this nice image. We can also add more detail by adding things like ambient occlusion here, and that will add more shadows and make things look more realistic. You can see it's starting to add in shadows here rather than before. So again, it's all integrated things that can quickly make your images look more realistic. So now we'll move this light up, and we can also add cameras. So you can have different kinds of cameras like wide angle or this classic lens. And I think I'm going to select the wide angle lens. That's what it kind of looks like. So it's making it more like a movie. And really you're kind of directing this scene here. So let's see what that, we're going to render out this whole animation of this thing falling. And I'm going to speed it up for the sake of time. And we'll be able to see it here. So now we have shadows and things are bouncing off of each other, and it looks pretty cool and realistic. And then again, this is just another example of rendering. You can see this is kind of bland before, and then as we render it, you get these nice, beautiful images. We can also mess around with materials to make things look more like hydrogels in this case. So next, something that's really interesting is to play around with the properties of these objects. So, right now everything is kind of this rigid body. So whenever we simulate gravity and we just drop this box, we can see that it just kind of does not deform at all whenever it hits that surface. Just like a rigid body would. But if we add in soft body physics, which again is just a simple right click and add a soft body tag to this cube, which I'm doing here. I'm sorry that I'm not showing all the details here. It just would probably take too long. But now as this soft body hits, you're going to see it deform. So super cool, way more realistic. You can see it deforming on the top here. So you can imagine this being like a soft hydrogel. And we're starting to get things that look more realistic now. And we can, here I'm going to go in and change the stiffness. So the stiffness was at 0. We're going to change it to 100. And this is the problem I have with this software. It's just kind of relative values. We can't type in, 200 kilopascals. But you can see that now it's more rigid and it's kind of bouncing around. So I think other software like Blender or even this software Houdini, you can actually add in real values. And I'm sure, FEM software would also be able to give you more accurate representations of what's the physics here. So the last thing I'll show you is we can actually play with friction. So if you drop this cube onto this surface, it's not really sliding off, but we can go in real time and change the friction, make it 0 here. And then as we go to hit play, this thing's just going to slide off because there's no friction anymore. So pretty cool stuff, and it's fun, a lot of fun to play around with. I don't know how to get out of this. Here's again, just playing with different stiffnesses of different spheres. So, it's a good way to represent things in a qualitative manner. We are also very interested in like soft particles and simulating how those pack together. So here's some mini soft microgels that we're packing into this container. We can see them all deforming on each other. And this does take a lot of computational power to do. So that's why I'm not able to do it. But one cool thing we've been able to do is actually simulate this packing, change the properties of these spheres, and then use the software to go in and look at the porosity within this structure. So you can kind of take Z slices of this object and look at the interface between particles. So this is actually something we've been able to use experimentally to understand how particles pack together. And I think this software is pretty powerful at running these simulations. So next, I'm going to show, so this is just something else, that can kind of highlight an object using that camera that I showed before. You can actually change the f-stop in that, which is, if you know anything, which I don't really know much, but I know that the f-stop allows you to kind of have things in different focal planes. And this allowed us to kind of have this complex environment with all these fibers. We can bring this one fiber into focus. Here's a really nice example of some that I like to show in my presentations, comparing a cell with natural ECM and electrospun scaffolds, which that's most of my research is on. And you can see by making these schematics that we can really highlight the difference between our scaffolds and natural ECMs and how a cell might perceive these environments, or these different types of synthesized materials, microfibers, nanofibers, and how a cell would experience or interact with those types of environments. So it's a really good teaching tool to explain the types of materials and the work that we do. Here's an example. Andrew Daly created this method to pattern cardiac spheroids and create cardiac tissues in 3D space. And I helped him by kind of creating an animation that explains, up here in the top right, how we're actually doing this. So I think, here he's showing one 2D image, and then I'm able to add to this information by showing 3D what is actually going on while he's showing these images. So I think it really adds to the impact of the work to show what's actually going on. We just place those spheroids, and then that's how we were able to build tissues. Another example I showed previously during a presentation here at CEMB on this recent work that we had accepted on these fiber assemblies. So using all the tools I've shown you so far, I was able to make this animation here, and I integrated it with some real data using another software called Adobe After Effects, which is essentially like video editing software. So we created these fiber assemblies by creating fiber particles, and then we photo cross-linked them together. So I'm using the light tool here to show this photo cross-linking. And then some of these more dynamic tools to show 3D printing of these materials and how they shrink under cell forces. Or if we print contractile and non-contractile filaments next to one another, we can control the direction in which they bend. And again, this is all with Cinema 4D software. We're able to simulate liquids flowing and photo cross-linking of those polymers into constructs, and how, this is just some more real data from that experiment. So again, this was a great tool to put on Twitter and help people understand the data without having to dive all the way into the paper and just quickly see what we did. So I think this is super useful for anyone who's on this call. And what these OBJ files allow you to do is just quickly create objects that are quite anatomically correct, and other artists have just freely added them online. So you can go to one of these software or one of these websites like Free3D, look up OBJ files, and then you can find objects like this car or these people and drag them into Cinema 4D. So I found this syringe because I needed that for that previous figure. Someone had made this and they just gave it up for free online. I downloaded that OBJ file, and then I drag it in the Cinema 4D, and you have this perfect syringe, which I would have failed at making if I tried to make it on my own. But the software just creates it very easily, and then we can change the colors and get something that looks really nice, like this syringe that kind of explained this process for making these materials. I'm just checking the chat. Shrinking in this software. I'll answer the questions at the end, but I was just making sure there wasn't anything right now that needed to be answered. So the other really cool thing and useful for probably everyone on this call is you can take Z stacks of your confocal Z stacks, and you can take each slice and turn them into OBJ files. So you can just have one channel open, save it as a .obj file in Fiji, and then you can drag that OBJ file into Cinema 4D, and it'll create a 3D rendering of your object. So here's a micro tissue that we created, and I created a channel for the actin cytoskeleton and the nuclei, and I dragged both of those into Cinema 4D and overlaid them. And you can see I get this really cool representation of a microtissue in Cinema 4D. I think you can also use software like Imaris to do these kinds of things as well. But I think this is a great tool for anyone who's using confocal microscopy to create some really cool, accurate schematics. So I'll quickly go over some useful plugins. So plugins can be expensive, but what they essentially are added software to the base program that you're using. So they're add-ons to do lots of other things that your software were never able to do. One of them, EPMV, which I can't remember exactly what that stands for, but it's a molecular viewing software, and it's completely free. It was created by some scientists at UCSF. But you can create structurally accurate chemicals and proteins using this plugin. So I just download EPMV, and then it's on Cinema 4D. I open it, and then you can type in the protein name based on, you type it in here, and it's based on the protein data bank. So you can just search for whatever protein. I typed in spike protein, and it came up with this spike protein. I type in 6JX7 into the software, and I click fetch, and then it brings up this perfectly rendered spike protein from this virus. And it not only does single proteins, but it can create arrays of proteins to create things like an entire SARS-CoV-2 virus. So this is how people are making these kinds of animations or images that you see all over media. And it really requires, all the work is done by the scientists who figure out the structure, and then the software can just create these things very easily. They actually just released a new package called CellPack, where they were able to recreate an entire bacterial cell and all of the proteins in it and how they're organized. So very cool stuff that's emerging from all of the cryo-EM work that's been going on. And this was developed by Ludwig Onten and Graham Johnson at UCSF, I believe. But they've been working on this for years. We're also interested in hyaluronic acid, so you can look up like polysaccharides on the protein database. I don't know why it's on the protein database, but I'm glad it is. So we can create HA, molecularly accurate, and incorporate this into like a fiber. I didn't really like this image, but it's just an idea of a way to do it. There's other plugins that can create nice images, render plugins for Blender. Arnold is a great one to use that I've seen a lot of scientists using. And then one I really like is X Particles. So here, X Particles is basically just a particle simulation software, and you can create lots of particles, and it doesn't slow down your computer too much because a lot of the other plugins can do that. But here we're kind of simulating 3D printing at different speeds or different fiber densities. And we're able to print them, see how they're kind of organized at different speeds, and go in and look at the structure. So here I'll just show a video of that, of the printing process. So we can kind of change the density based on the speed, and we can look inside of that filament using this animation software. So very cool way to represent what we're doing. And last thing I'll show you is just some sculpting. So this is something I've recently been getting into, but we can create this sphere and then go into the sculpting setting here, kind of create more voxels here to get more definition while we're sculpting. And we can start to sculpt something that looks like a cell. So essentially what we're doing is using this grab tool, we go in onto the surface and literally just pull out strands, and you start to sculpt this thing into something that might look like a cell. So I'll just kind of speed that up. We can add symmetry so that things, the cell becomes more symmetric with its projections. Maybe these are Philopodia or something interesting like that. Or maybe this is a plant cell, who knows? Definitely don't know enough about plants to be saying that, but oh well. So we can also use this knife tool to add more definition. So we can start to sculpt out different regions and make this thing look really interesting. Kind of looks like leaves, so that's why I said that, but that's pretty dumb because leaves are made up of multiple cells. But anyways, it's a very interesting technique. I think it's a little more complicated because you have to kind of sculpt this thing like a piece of clay, but it can give you things that look a lot like the cells that we all work with. So I sculpted that, added a material, so it's green, and then we can render it and get this nice, pretty image. There it is. And then, some example where this could be useful is mimicking development. So here's a simulation of like a single cell becoming multicellular and then undergoing gastrulation and then extension with all these other, I just kind of made this up, but I was trying to mimic what we see in zebrafish development. So I think this is a super cool animation, a cool way to visualize things using sculpting. So with that, I'll just thank the Burdick Lab for listening to all these crazy ideas and, letting me work on their projects with them, making some of these types of schematics and helping highlight their research. And of course, for Jason letting me explore this in his lab, even though we normally make materials. And if you're interested, you can check out my website. It's bioshapes.myportfolio.com or Twitter if you want to interact with me on there. So with that, I'll look at the chat and take any questions. And I'm just gonna stop sharing, I think. Okay.
Thank you, Matthew, for presenting. I think in the chat channel, we have at least 3 questions.
Yep. So I'll go to the first question. Yes, it is very cool software. Thanks for watching. It is complicated to apply shrinking in this software, or that's a question. Is it complicated? It is not. You can actually, using the soft body physics, you can just change the volume or the length of your object and program it to change over time. So you can actually, it's very simple. It's just a percentage of the original value you can set to 50% and the object will shrink to 50% of its original volume. So that's fairly easy, although obviously it's not accurate for modeling and things like that, but it's a great way to show if you want to show someone shrinking with your research. Is it possible to simulate the motion of molecular assemblies in nanocomposite materials as a function of possible molecular interactions? So this is something we've been interested in with like spheroids and microgels and because we see them kind of stick to each other and we'd like to simulate that interaction between particles and cells. But I don't think this is a good way to model that. However, you could mess with like the friction of the molecular assemblies in your nanocomposite materials and potentially use the friction between particles to kind of simulate something you've seen through your data. But I don't know about the resolution. This is all relevant or relative to whatever particle size you use. So that's hard to say. But I think, yeah, you could make something that looks like what your data is, what you're trying to represent, but I wouldn't make any like conclusions from that. Um, Two questions from Mohammed. How do you create the fibers? So I'm guessing you're talking about in the software. So to make the fibers, we'll just create cylinders, and then I add different random effectors that will kind of bend and cause fluctuations in the fibers, similar to what we've seen through experimental data, kind of based on the persistence length. So that bending tool is very useful, or you can just simply draw out shapes that are similar to what you've seen. And how physical are the packing simulations? I don't really know. I would highly recommend looking at CellPack and AutoPack. These are softwares uh, that simulate packing on the molecular scale, and I have not messed around with those, so I don't really know how accurate that is. With our larger materials, like packing of microgels and things like that, it looks a lot like what we see experimentally, and we get similar porosities. But if you look down at the single particle level in these systems, you can see that there's actually a gap in the software. They don't physically let them touch, whereas we know that they're touching in our experiment. So that's one limitation. But yeah, I think, it's what I'd like everyone to take away is that we can start to really explain and communicate our science really well with these types of things. But I've struggled a lot with trying to get useful data from them. But other people in our lab have gotten very useful data. So I think you just have to be creative in how you use these tools.